Right. Welcome everyone. Good morning, good afternoon, good evening. Welcome to today's webinar hosted by the International Society for Biopharmaceutical Statistics, ISBS. My name is Dong Xi. I'm from Gilead Sciences, and it's my honor today to be your host today for this wonderful presentation from Costas. Let me introduce you the presenter today. Costas is a member in the Advanced Methodology and Data Science group at Novartis, where he specializes in exploratory analysis for biomarker discovery, digital biomarker development, and assessing treatment effect heterogeneity in clinical trials. He earned his PhD in machine learning from the University of Manchester, and afterwards, he worked as a postdoctoral researcher with AstraZeneca and Roche. So thank you very much, Costas. And I'll turn the floor to yours. Thank you.
Yes, thank you very much, Don. I hope you see my screen. I cannot see the Zoom call. If you have any, if something doesn't go wrong, or even if you have something you want some clarification, feel free to interrupt me. So I will present you the work that we put in place the last years in Novartis on a workflow for assessing treatment effect heterogeneity. And before moving on, I would like to mention that this is a joint work with for other of my colleagues in Novartis, and we have some group of collaborators and co-authors. And I'd like also to thank the FSPI Treatment Effect Heterogeneity SIG for the nice discussions and feedback in this work. This presentation will split in two parts. The first one will have been some general introduction in the workflow that we have available in pharmaceutical statistics. And the other one will be one implementation of the workflow that builds upon some ideas of causal inference. And we put the paper in archive a few weeks ago. So let's start with this imaginary scenario, which I borrowed from one presentation of Professor Richard Hamworth. And let's imagine that we have a trial that I failed. And after the initial, let's say, shock, the team was trying to find some to understand better the data, and there is a person in the team that he's very good in going deep in the data and the tables, and he finds that for women over 50 the response rate is 70% in our drug and 30% placebo. And of course then they start all of these discussions, is this a true finding, or they can be even more ambitious, that maybe we can go on with this subpopulation for another trial, or we can do some claims around this subpopulation. And then it comes the statistician or data scientist that start feeling nervous around this, and clearly something not exactly right is going on in this direction. And I remember the first time that I also engaged in this type of analysis, I was feeling this nervousness, but I couldn't clearly explain what was the origin of this nervousness inside me. So with this type of work, we try to build some workflow to have a bit more confidence in our analysis and contribute in this type of situation more constructively. Now, before moving on, I want to say that I will use the term subgroup analysis and assessing treatment effect heterogeneity interchangeable. And this problem is quite difficult, and this, let's say, widely acknowledged. And this nice paper from Steve Ruberg that he even emphasized this in the title, that assessing and communicating heterogeneity of treatment effect for patients' population is the hardest problem. And at least in my, simple understanding why this problem is difficult is a combination of two things. We are in one scenario that we have a high chances of false negatives because of the insufficient sample size. The clinical trials, they are not designed to, most of the time, to assess subgroup effects, so we don't have the power to do some strong claims. And at the same time, and more concerning, is the high chances of false positives due to multiplicity. And because we have a big space of possible subgroup that we can explore, we can have a lot of, possibilities. And then if we go on with the best, the subgroup with the highest treatment effect, without accounting for all of this big subpopulation we explore, it leads us to this area of finding something that we cannot replicate later, and this area of selective inference. So now I will try to keep motivating the difficulty of the problem with this toy illustration. We have, let's say, even within the RCT setting, we have a phase 3 program that it was two twin studies, and it's quite big, the sample size of 600 patients. And actually, this is actual trials. The endpoint is rate reduction, so the lower the better. And we initially said, okay, let's consider the subgroups that they defined by one baseline covariate, and there were 70 subgroups that we can define with one covariate. And here you can see the treatment effect in study one for these subgroups. This is the estimates, the point estimates of the treatment effect. So you can see that there is some variability around the 0.76, which is the overall effect in the study one. Now, when we plot the treatment effect in the study one versus the study two, ideally we'll want to find whatever we see in one study to see the same in the other study. So let's imagine that here we have this subgroup with a treatment effect of 0.6 in the study one. Ideally, would have liked to see the same effect in the study too. But what we see in this case is that there is no strong treatment effect in the study two. Actually, there's no effect at all for the same subpopulation. And there is no modeling, there is no, let's say, modeling to derive the subgroup, there is no machine learning or anything. It was just subgroups of one that they defined from one baseline covariate. When we start defining subgroups with two baseline covariates, this correlation is even weaker. And you can see in this scatter plot nicely captures this. And while this maybe motivates some side of the project, there is the other known issue of subgroup analysis that is very well also reported in the literature that they have to do with these false positives and these dangers. And this can lead to findings that you cannot replicate. And there is this paper of Yusuf et al. Back to 91 that they identify 9 papers that they were doing some subgroup claims. And for all of these subgroups, there was some attempt to confirm it with another trial. And there was no clear signal in any of these attempts. So, and this is quite an old story back to 91. Even more recently, Professor Ioannidis and his group, they had a bigger study that they identified 64 RCTs with 117 subgroup claims. Out of these 117 claims, 46 were with some statistical significant test of interactions. So 46, they have statistical significant, let's say, heterogeneity. From these 46, there was attempt to replicate through meta-analysis or RCT the 5, and none of these 5 they managed to find any signal. So this actually captures nicely why we have this nervousness when we end up in this type of discussion around subgroup claims. And the high-level message that without external replication or plausibility, purely data-based findings can be very speculative. But so far I was discussing about why this problem is very challenging. At the same time, it's very important, and a lot of internal decisions rely on the this type of analysis, and this analysis can influence decisions around enrichment, around generating hypotheses for new trials, and also a decision on how we can prepare towards health authority questions. And this is also very well appreciated externally from the regulators that this is an important problem, and there is guidelines and white papers. And what I like a lot is the quote from Rob Hemmings from this last paper that says that ignoring the problem and similarly routinely dismissing results of subgroup analysis is not a scientific solution. So we need to have in place some solution that we can trust, we can do the best that we can, and provide this analysis. Just now, a summary is that data alone, they don't speak for themselves when the question is around treatment effect heterogeneity, because 95% of the trials that we are planning, they are not tailored to answer this question. So we need some external information, which can be take a lot of forms. It can be historical trials from similar drugs in same indication or same drug in other indication. We can have preclinical information, mechanistic understanding, and of course, some biological plausibility. And without replication or plausibility, database finding alone is very speculative. So this was the motivation behind our workflow. And here it's how the workflow looks like. And we gave this acronym WATS because I will come back a bit later also, another motivation of the wording WATS. So it's the main aim of the workflow. It's to generate insight on how treatment effect varies with baseline characteristics. We would like this to be a pre-planned analysis and the results to be quickly available after the database lock. Our ambition is to combine external and data-based evidence to improve decision making. And we believe that a systematic approach that fosters transparency, reducing misunderstanding or errors, will allow in a better replication, will naturally lead in a better replication of our finding. In scope, it's assessing treatment effect in phase 2 and phase 3 trials. And out of scope, it's confirmatory statements on subgroup findings and subgroup effects. So as I said, the workflow splits in 4 steps. And now I will dedicate some slides to describe a bit in more high-level detail each steps. And then we'll go to a bit more technical solutions on how we assess the treatment effect heterogeneity. So in the analysis planning stage, we want to involve and align with the stakeholders and try to discuss about what is the question that we have, what is the outcome variable that we are interested in, which is the scale, which is the treatment effect heterogeneity we are interested in, which scale and with which type of comparison. We want, treatment effect of the drug versus placebo. A lot of times maybe they were interested in heterogeneity across different active dosages. What studies, if there is more one study, what studies to include and how to perform this pooling. Then the more important thing for us is also to identify the baseline variables to include in the analysis. And here we would like the teams to not only to tell us what variables to include, also to sort of to document the a priori evidence that these variables can act as an effect modifiers. And we want this to document it in the analysis plan, and we provide some guidelines of how to choose between these 4 levels that we have: the non, low, moderate, and high. And this can take some tables and missing data and so on. Now, in the second stage, we start now working with the data. Firstly, we want to understand our data without approaching the main question, which is the TH. So we want to see some univariate summaries, what is the dependencies between the data, and we follow this IDA, this systematic workflow that gives some sort of rules on how to work with the data responsibly. And our colleague Mark Bailey had a nice paper with a Stratos initiative around this type of rules. So the outcome of this initial analysis, there are some visuals that we can see and try to understand the data in many characteristics of the data, distributional characteristics, missing values, if there is skewed in the categories, if there are rare categories, if there are highly correlated variables, and all of that will understanding will lead us to take some decisions how to pre-process the data before touching the analysis question. And this is some example decisions that we can take, like, transform baseline variables, exclude variables with high missingness, merge sparse categories, because, many times having some category with very few examples will not useful neither for the method, but also it doesn't provide a lot of insights also around these questions that we want to answer. We want to choose between highly correlated variables, and here again we try to get the input from the clinical teams to how to, if they have some interest in some variable versus the other. And of course, we want to impute the baseline variables in one hand, but also the outcome variables. And then we try to follow always the primary analysis, the estimate strategy. So after this step, now we have let's say, a complete data set that is very suitable to fit to statistical and machine learning models. And we move to the core part of the analysis, which is the exploration of treatment of heterogeneity. And to this point, building upon also some work that we published, that we benchmark many methods in the literature, we approach this problem in 3 levels. So we have 3 questions. The first one, we want to have an overall evidence against homogeneity. So we want to answer the question, how strong is the overall evidence against the null hypothesis of homogeneous effect? The second one is, which are the baseline covariates that drive the heterogeneity? And the third one is how the treatment effect changes with the identified baseline variables. And now we'll go to each of these steps briefly. As I said, in the first step, we want to see we have a global hypothesis test against the null hypothesis of homogeneity, and which we feel that maybe it makes sense to have this null hypothesis because the trials, they are designed with some specific population in mind, and so there is this implicit expectation that the effect in this population is homogeneous. And we use some global tests that also penalizes for the a number of variables that you include to the analysis. And the other thing that we want also to emphasize is because this is exploratory analysis, the p-value derived by this test, we want to approach it not in the dichotomous way to take binary decision above and below some specific threshold, but more as a continuous in the continuous scale. More as a measure of divergence against the null hypothesis. And at the same time, in the communication with the team, we want to emphasize maybe a different way of interpreting the p-values that is motivated and introduced by this paper of Greenland and collaborators, that they approach the p-value as a measure of surprise. And I like this example. So, for example, if you see a p-value of 0.063, seeing this, it will be equal with a surprise that we'll get if you toss a fair coin and 4 times you see heads, let's say. Or if you go 0.001, it is 10 times you see heads. So you see, 4 times heads, maybe it's not that rare. 10 times it starts getting very rare. So it's also the interpretation should also depend in the context. But we believe that this parallelism with this throwing a coin, it may help a bit for us to get a bit of a better understanding of this distance, let's say, from the null hypothesis. And we provide some verbal wording also on how to interpret these p-values and some arbitrary, of course, some levels. Then in the second step, we want to identify the effect modifiers. And these effect modifiers will help us to identify some populations of heterogeneous treatment effect. And to this end, what we are currently do, we derive importance scores from a multivariate modeling that captures how strong each variable act as an effect modifier. So we provide, again, all of this to the teams, these visuals, and then we ideally we want to discuss and understand how plausible is these findings. And finally, we present how, with various visuals, how the treatment effect changes with the identified effect modifiers. And all of this visualization, we want to be the starting point for the discussions around heterogeneity with a clinical team. And this comes to the multidisciplinary assessment. So also this was one motivation behind choosing this acronym codes, that we provide a lot of visuals and want the team to watch them and then start to assess the credibility of the finding. And always we want in this assessment to take into account what we have documented in the planning around how plausible these baseline variables can be as an effect modifier. So we have the a priori evidence, and we want to combine with what the data provide, the data insights, and always we encourage to avoid strong confirmatory language and to have this open discussion in the context of what we were documenting in the planning. And in a high level, the main purpose is to interrogate the data and generate insights. What comes next depends on this insight and, of course, the situation, every specific situation. So here is some things that, from my experience, is the next steps. A lot of times from this analysis, you find some findings that the team it's interesting to explain, so you can go deeper to provide some insight around the findings. A lot of times they come with new baseline variables or new endpoints that they would like to explore the heterogeneity. A few times, they are happy with the analysis and then there is no follow-up. And again, not so often, maybe the team is interested in the treatment effect in a specific subgroup. And then, of course, we need to use estimates that they are corrected, let's say, and can be stringent type of estimates or Bayesian type of estimates to derive the subgroup effects. So now, as a summary of this first part, it's one workflow that provides a systematic approach to explore the treatment of heterogeneity, considering external evidence and the best scientific understanding. This is an ongoing internal project, and we have engaged with various trials at the moment, and we use this tool to answer different types of questions. The main starting point is to identify populations of differential treatment effect, but as I said, a lot of times is to identify a population that respond better to one or the other arm, high versus low dose. Let's say we can use it for identifying population that manifests specific adverse events, and also you are trying to use it to explain multi-regional difference. And in the E17 guidelines, already they emphasize the importance of deriving predictive and prognostic markers to explain the MRCT trials, the findings from the MRCT trials. So this summarizes the first part of the presentation, which was to discuss the general workflow for assessing the treatment of heterogeneity. And now I will move to one implementation of the workflow that is suitable for continuous and binary endpoint. And it builds upon the idea of using some causal inference and machine learning tools to derive the individualized treatment effect. Now, again, this is the workflow, and we will focus now in these three questions. We want, and now in this slide, I will try to communicate in the high level how this method will work. What's the driver behind this method? Let's imagine that we have a population in our trial, and we split it in two parts. And half of the population takes the our drug, the red drug, and the other half, let's say, takes the placebo. So the primary analysis focuses on the average treatment effect across this population, and this can be take this form. What was the expected outcome in the drug minus in placebo across this population, the expectation. Now let's imagine that we're a doctor in the clinic. It tenders one patient. Maybe at this point we are not care so much about the average treatment effect in the population, but we would like to find what is been the average treatment effect in the person with these characteristics. And a popular estimate, it's the conditional average treatment effect, which is the expected outcome in the one in the drug minus the placebo, given the X this time. So this way we derive something that captures a bit more individualized effects, and this is the driving force behind our methods. And now, before moving on, a bit of some terminology. We'll have X1, X2, and XP. We have P baseline covariates, the outcome Y is our endpoint, and we have the intervention Y equals 1 and Y equals 0. And as I said, in this setting, together with Y, they come to other outcomes that they are not fully observed. The Y1 is what was the outcome if the person was taking the drug, and the Y0, what was the outcome, what would have been the outcome if the person was in the placebo. And ultimately, we're interested in this contrast per patient, Y1 minus Y0. And this is not observed. And this is what it's called the fundamental problem of causal inference, that you observe one part. And on top of that, what we are interested in is to explore how the X, the input space, interacts with this Y1 minus Y0. For example, we want to see what are the drivers in the X that influence the this individualized treatment effect. And this will help us determine, as we said, how well the patient's response to the treatment. So this is the quantity of interest. But before moving on that, there is a big literature on how to estimate the average treatment effect. So there is this method that is known as a Z computation that relies on the outcome modeling. In this case, let's imagine that we train two models, one with using the population that got the treatment, and one the population that got in placebo. And then for each patient that makes some predictions, we take the average and we summarize across the patients. A different approach is this inverse probability weight, which this time we model the propensity score. So the model that we will now it's to predicting the treatment allocation given the axis. So these three boxes are different models to derive the average treatment effect, and then it's the augmented inverse probability weight, which is a doubly robust estimator, which combines the IPW with something that is built around the outcome model. And the double robust estimator has two nice properties. The first, it's consistent if either of the outcome or the propensity model is consistent, but also it's more efficient because the more if you have well-specified models, this improves the efficiency, and we'll see it also in the simulation. So this was the expression for the average treatment effect, and there was this paper that we built upon of Kennedy that he was using the summand, the each individual term, and he was making us the pseudo observation. So for each patient in our trial, we can imagine that we can derive this term using these three models, and then he was regressing over the against this pseudo outcome psi on the axis to derive the case. So essentially, we have another model on top of this pseudo observation. Of course, he was doing this in a very nice way with applying a lot of ideas, and I will see the algorithm later. And the main point was that for each patient, we derive some pseudo outcome, which can be seen as a proxy of the individualized treatment effect, and then we do this final regression. So this is the algorithm that he introduced, and there is it builds upon the idea of this cross splitting, which can be seen as this cross validation. So we take the data, we split them in different folds. We use the training data to train the nuisance models, this the propensity score and the outcome regressions, and then using the prediction of these models, we derive the pseudo observation, and then we use the test data to train the Kate model. And then we repeat the same process against all of the faults, and we derive this algorithm derives two things: the pseudo outcome and the Kate. The initial, the Kennedy focused more on the Kate and all of these properties that these Kate estimates have. But in our work, we thought that maybe we can use the psi as a way to answer all of these three questions of heterogeneity, not only to estimate the Kate, which can be seen as the third question, but also to use it for a global test and also for deriving the drivers of heterogeneity. So a bit before moving on to our implementation of GOTS, we want to emphasize again this algorithm essentially builds four models and with different data every time. Now in our work we use also this idea of stacking, which is an ensemble of models, and we introduce in this ensemble two different types of modeling paradigms. One is the regularized regression, and the other is something that builds upon random forest. And what this super learner is doing, derive some optimal weights, and optimal in the sense that they minimize the cross-validation prediction error. And the final prediction of the model is a weighted combination of the predictions of the individual models. So the main idea is that it leverages the strengths of the different models to improve the overall performance of the final model. So now we use the algorithm of Kennedy for the DR learner, and for its different models, we use the super learner to train them. And after all of this procedure, we have this data set, which is the baseline covariates. And now this time we have also this proxy of the pseudo outcome we can see as a proxy of the individualized treatment effect. And let's say, okay, now this it's what we call machine learning. It's a supervised machine learning dataset. The dataset that you can apply any regression technique on top of that to answer whatever question you have in mind. So now we use this dataset for all of the steps of the GOTS workflow. And the first one is we want to test independence essentially between the multivariate input space X and the psi. And we phrase this null hypothesis. And we want also these p-values from this model to have two properties. The first one is that when there is no heterogeneity in the data, we want the p-value to be uniform distributed between 0 and 1. And when there is heterogeneity, we want the p-value to be as small as possible to have a powerful method. Now, to test this independence, we use the permutation-based procedure from a package of the coin package from Hawthorne et al. And this package also gives you some flexibility in terms of the statistics that you can use. So the two main statistics that we explored was the maximum statistic that you can imagine that for each baseline covariate, it calculates a test statistic, and the overall it's the maximum. So this is suitable when there is a single driver of heterogeneity or very few. And the opposite is the quadratic statistic, where all of the statistics of the features collectively define the overall statistic. And then we had some benchmarking simulations that they imitate the real trial, and we provide details in the paper. And we explore how these two different statistics, the two different methods for testing the heterogeneity perform. Firstly, in scenarios that there is no heterogeneity, and always the methods was providing a nice uniform distribution from the p-values. This you can see that they will control nicely the type 1 error and adjust it, of course. And then when there was heterogeneity, we explore scenarios. And this specific scenario that I showed you is the scenario that only one variable, the x14, drives the heterogeneity. And we can see that the maximum statistic, as we were expecting, performed better than the quadratic statistic. And we have more simulations in the paper. Then we move to the second objective, to derive the effect modifiers. And again, now we have this dataset X psi. It's the traditional dataset that we do for regression. And we want to rank the x's according to the importance toward the psi. And we have, again, one desirable property. We want the procedure not to be biased towards different characteristics of the features. And mainly we focus that we want the procedure not to be biased towards continuous versus categorical variables. And this is something that is uh, well known to some models, like the default random forest. So let's imagine that you have two variables that they have the same content of information, and one it's continuous and the other it's binary. The method will pick more often the continuous to do the splits, because it gives more degrees of freedom to provide splitting that lead to more pure results. So now we explore two different ways for deriving the effect modifiers. The first was to use a permutation importance score on top of one version of the random forest, which builds upon the conditional and inference trees, that by design tries to overcome this bias. So it's sort of unbiased between continuous and categorical variables. And the other method was to use some new method for deriving importance scores introduced in machine learning literature. And it's motivated by some game theoretic principles, the SAP values, but this time around of the traditional random forest. And again, we simulate two scenarios, one without heterogeneity, and then you can imagine these methods take the data, there is no signal in the data, and they produce a ranking. And then we repeat this many times. So we want these rankings to be completely at random, because there is no signal in the data. So what is the first variable? We want to be, every time to see something different, not to be some bias in the top hit. So what we see here with a different uh, symbols, the circle is the categorical variables we had in the dataset, and the cross is the numeric. So when we use the random forest as a model, constantly the categorical values are having less chances to appear in the top of the list, which it shouldn't, because all of the variables are irrelevant. And this is not the case for the sea forest model. And also then in this scenario, we had two variables that they were deriving the heterogeneity. And we have now some heterogeneous effects inside the data that we simulated. And in this setting, we saw also that the sea forest is more powerful. But of course, it's not always the case. In some other simulations, maybe the Other method was performing a bit better, but overall we chose the C4 because of this characteristic that is not biased between continuous and categorical variables. And for us, it's important because in the trials we have always in these assessments that we do, it's mixed type of data. It can be some markers, can be some categorical variables like gender, ethnicity. We don't want to be biased. And the final objective that we had was to estimate Kate. And again, the paper we have some many simulations. Here I will show you two graphs. In these graphs we compare the different methods, the DR learner against its main components, which the IPW and this T learner, which can be it's known also as the virtual twins in pharmaceutical statistics. It can be the counterpart of the G computation in the learners. So we have three methods. We see different levels of heterogeneity. In the y-axis, we have the mean squared error. The lower, the better. And we can see that the DR learner it's always more efficient than the other two methods. And also there is a big literature and a lot of different ways of performing the cross-fitting. So here we chose two, the one that originally was in the paper of Kennedy and another one which is motivated from some other methods. But again, we could see that one method was always performing better than the other. So we chose this way of doing the cross-fitting. And then we compare against some other alternatives that we had also in this neutral comparison paper. And we could see that the DR learner, it was performing nicely. Always this is quite a difficult problem, a lot of different objectives, a lot of different modeling. In a high level, the DR learner, there was no scenario that it was performing really bad. Maybe it was in the top 2 or 3 of the methods. Sometimes, of course, often the top 1. But it's overall, it was one way that we thought that it's a nice to move on. Also, it has nice characteristics in terms of how to interpret and how it have some simplicity inside. Okay, and then I will show you some application of the DR learning in some clinical trials. Here I focus in the disease of psoriatic arthritis, and quite a nasty disease that manifests in many areas of the body. Novartis has one successful treatment for adults patients with active psoriatic arthritis, and we focus in 5 phase 3 trials. So we had quite a big data set around 2,000 patients. The primary endpoint is a composite endpoint, which is at week 16 in a high level. When y equals 1, the person is responder. Y equals 0, it's non-responder. And our estimate is the risk difference. It's the probability of being responder if you are in the treatment minus being responder if you are in the placebo. And in this analysis, we include 70 covariates. And as said, these covariates is mixed, categorical and numerical. And they were demographic information, medical history, lab values, clinical measurements, quality of life assessments, efficacy measurements, and one proteomic marker. And there was already 3 papers from Novartis authors analyzing this data with a different model links and different inputs. So one of these papers was building upon some idea of generating some fake variables and then feeding in the model the original, the fake, and by knowing which are fake, you can provide some selections that they control the false discovery rate. And in this paper, these red were the variables that they selected by the method. Then it was another paper that actually it's completely different. We ran an internal challenge within the company 2-3 years ago, and we gave this data to many teams across the organization. And after 2-3 months, they perform analysis and they gave us what they believe is the subgroup of enhanced treatment effect. Here in this graph, you see how often each variable was used across this submission of the teams. So the first 2 variables, the C-reactive protein and the H, are the same with the ones that we have found in the different paper using different modeling. So it's interesting. And this, the third variable, the fatigue score. So it's interesting that there is some consistency in the top of hits, which is the C-reactive protein and the H. And then there was a paper from another part of the organization, a bit more research that they perform a thorough analysis in some proteomics markers. They analyzed 4,500 proteomics markers, and they found that the one marker, the BD2, at the baseline, can be used to define, responders essentially in the treatment. Can use to define heterogeneity. So what we did now, we include all of these clinical variables plus the this proteonomy marker, and we want to see the new method that we suggest that builds upon this idea of the dear learner and the super learner, and can identify similar findings in these datasets. So first of all, the overall assessment gave a very low p-value, which it was a good sign because we already knew from the literature that there is some heterogeneity in this data. Secondly, we use our method with a dear learner and then the rankings coming from the C-forest and the permutation. And this is what is the outcome of our method. And we can see again in the first two hits, it's the C-reactive protein and the H, which it's exactly the same with the other two papers that were doing different modelings or the subgroup challenge. And then the third hit was the proteomic marker, which is nice. So this also it was the first work that put all of this into one. And then again, we could see the fatigue score, which again, the papers that they were focusing the clinical variables was in the top of the hit. So this also shows that, okay, this method can provide something sensible. And then, of course, we could use this method to derive subgroup estimates. And in this graph here, we have these three pre-specified levels of the C-reactive protein. And with blue, it's the overall treatment effect in the trial. And then we have with black dots is the observed treatment effect in this subgroup. And the star is the estimate of the treatment effect from the Kate, from the dear learner. And you can see that also this method nicely sort of provides some sort of method that shrinks towards the overall efficient goal of the data to build the models. And this is again the same graph, but this time for the proteomic marker. Now, as a summary, we use a non-parametric and robust modeling building upon the dear learner in combination with a super learner, which is an ensemble, a stacking procedure motivated from the machine learning to derive for each patient a pseudo outcome. Then we use this as the driving force to answer the three questions that are important for the WOTS workflow to assess the treatment effect to heterogeneity. We apply this method to some psoriatic arthritis trial, and it performed nicely. And overall, we believe that this double robust version of GOTS provides a nice way to explore the heterogeneity when we are interested in continuous or binary endpoints. And before finishing, I can mention that the previous week we put also in archive one of paper that we try to provide the same answers, but this time for different types of endpoints like odds ratio or hazard ratio using parametric model as the driving force. So yeah, thanks a lot for your time, and yeah, I hope I was on time.
Yes, thank you very much, Costas. You're perfect on time, and I think we do have some time for Q&A discussion. I want to check from the chat if there's any question. Not so far. I think I can start to ask maybe one clarification question. If you go to the slide where end of the slide deck where you show the observed effect and the kind of predicted effect. Yes.
Can you see it now? Yeah.
Yes. So here I think you have two plots where I do see the trend of that the DR learner treatment effects.
Yeah, can you see both plots now? I removed the animation. Okay, nice.
Yeah, so the DR learner treatment effects, they are closer to the overall treatment effect.
Yes.
Right. Which I think it's nice in the way that it kind of adjusts a little bit for the random deviation from the overall effect. Is this something that you expect every time we run this, or this is just
This is a very good question. You know, we haven't explored systematically, but through the practical applications, I haven't explored the properties of the estimator in terms of you know, bias and variance against other procedures. But at least in my mind, I explained this string is because of that you revisit all of the data and you provide some model. Of course, now it's as I mentioned, rarely we use this now for providing estimates. You know, maybe you can see it in comparison of more Bayesian approaches or other approaches which by design, or stringate approaches that by design perform these stringates. But yeah, this open for it's an active area that we try to understand a bit better these operational characteristics of the Kate estimate. To be honest, most of the time in practice, we are interested in the first and the second question. You know, the third question it's not the core. But yeah, I don't have any proof that by design should do these stringates, but by you know my experience in practice, and also how aggressive this stringates maybe. You know, you can try to understand how this.
Yeah, no problem. Yeah, thank you very much.
Yes.
Do see there's one question from the chat. Maybe I can read
It's a
it.
good point.
What sort of sample size do you need to have reasonable performance for this workflow and the DR learner?
Yeah, this is a very good question. Actually, the sample. I think the sample size. It's important also to decide how aggressively you should do the modeling. So now, if you have not a big sample size, maybe it doesn't make sense to do very fancy non-parametric baseline models in the super learner. So a lot of times we go more, you know, simpler models like linear regression or some additive models. But yeah, in this particular example, it was 2,000 samples, but in the simulations, we were trying to simulate scenarios from 500. So that was more realistic of a single trial. But this is also the nice thing of this framework, that it gives you flexibility to put the ingredients inside. So, you know, if the sample size is not big, I would have recommend doing less fancy modeling, more simpler modeling, and also maybe you should be ready to perform some sensitivities analysis in the findings.
Thank you. I hope that answered the question. There's another question from chat. For the DR method to detect treatment effect heterogeneity, shows the great capacity to detect the important moderators related to different treatment effects. Can be able to detect the cutoff points to identify higher, lower treatment effect based on a subgroup with specific cutoff points of continuous variable.
Yeah, I think by default, no. I would say no. There are some other nice methods that you know, Professor Richard Samworth and they have some methodologies of deriving the signature, let's say, of the sample subgroups, of course, by controlling some type 1 error and make some parametric modeling. But in our case, this wasn't the focus. So I think this method of the self, no. Maybe you can have some wrapper method when you create this regression problem. Then on top of that, there's a lot of methods for subgroup selection in this regression setting by controlling the type 1 error. But you know, we haven't explored that.
Got it. Thank you very much. Maybe I can add a follow-up question to this one, because I'm curious about, in practice, what should we do? Let's say if we have a covariate that we could treat it as a continuous variable or say a categorical variable, right? Sometimes we have a better idea of where to have the cutoff points. Sometimes we don't. So then in this case, should we put all of them into the model?
Yeah, I would. Yeah, what I do in practice, when I have continuous in the models, I put the continuous variables, and then in the visuals, a lot of times the teams also it's interested in the categorical versions. But at least in my mind, if you have the continuous version, maybe it's more suitable to fit it in the model. You know, you need to. But again, maybe it's a bit more empirical or machine learning. Then a lot of times you want to see that the results are robust. If I fit the continuous or categorical, and I see big difference in the findings, I will put in question the whole findings. But if you see some robustness, then it's always nice. But I tend to use the continuous versions for the analysis. Yeah.
I see. Yeah, because another question comes into my mind is if you put many kind of similar variables, I don't know how the correlation could affect the performance.
Yeah, this is also what we were trying to do. There is some methods that they shouldn't be affected. Let's say like the random forest, because with the splits, when you have some very correlated, you ignore it in the second split. When you do regression, then lasso it's getting affected. So we're trying to, but always we recommend that if there is a very highly correlated variables to take one decision before running the analysis, to choose between these two. And so we hope that this combination of models will help us a bit, but I think it's better to take it in this IDA phase, discuss with the team and try to see if they have one interested more in one than the other. And of course, also I forgot to mention we also provide some visuals on this interaction importance, which more how the variables are correlated within the model, not not let's say the correlation of x1 and x2, but sort of conditioning to how correlated they are. And again, this help with deriving some sort of joint spaces. But again, all of that can be seen as a starting point of discussion, but not something.
Thank you. Let me see.
Thank you. Yeah, all of the questions are very interesting. Yeah, I don't know if I have convincing answers, but some of them it's also open questions for me.
No problem. Thank you for your answers. Okay, there's another question.
Yeah, very good question. About can you provide some added explanation why you decided to combine lasso with random forest method in SuperLearner and not ElasticNet? So actually, yeah, we explore many combinations. Initially, we're also working a lot with XGBoost, and then we explore different types of random forest. At the end, we're using this benchmark simulations that we have to take some decisions, but the performance wasn't changing dramatically. Sometimes there was some combination that was very bad, but in terms of what is the best, you have some sort of together. There were some methods that were together. So this elastic and random first, I remember that wasn't much. The performance was very similar also with lasso. So our main driver was to keep the simplicity and having this benchmarking as a way. Of course, maybe someone can say, okay, this benchmarking maybe also a lot of times clinical trials you feel that this is very high, the signal to noise ratio is very low. So even if you spend a lot of build a very complex models, maybe they perform similar with more simpler models. So, and since the outcome was more the drivers, I think the evaluation measure for all of these methods we explore, the combination was similar. Apart of some methods that were performing a bit bad, like the XGBoost, if you don't optimize it, and there were some methods that they were a bit requiring a bit more optimization, which means more splitting and more time. But yeah, the lasso and ElasticNet was the random forest and ElasticNet was performing very similar. We explored lasso, ElasticNet, and READS regression, these three regularizers. But,
I see. Yeah, thank you for
thank
that
you.
question. I have another question. I think from my experience, it's important, as you illustrated, that to get some evidence before you look at the data to see what are the possible different sources of information to support the treatment effect heterogeneity, right? Including getting, say, some opinions from stakeholders.
Yeah.
Right. I don't know whether you could share some tips or experience how to best solicit their opinions.
Yeah. So this is a great question, and actually, it's a work in progress. Initially, what we're doing it was just documenting the variables, but then a lot of times you see that if you perform the analysis and you saw the results, everything may make sense. You can find
Yes.
everything or papers in the literature now. It's you can connect. So we're feeling that maybe it's important to document this evidence in advance. So now we run it in a couple of trials. This again, it's not that it's that it's a we have some clear roadmap, but I think it helps a lot, at least even for the clinical or subject matter experts to reflect a lot when they see the results on what they were uh, expecting, and not to jump directly to the graphs and build stories. So I'm not sure if I have a clear, because a lot of times this is also it have with the people. Sometimes you work and it's very smooth, and some other times it's very tricky. And to this end, also it's the p-value. Also, always we said you shouldn't use it as a binary decisions, but always the people also tend to interpret around 0.05, 0.2. So we have all of these ingredients, but we don't have the golden recipe now. But as I told you, a lot of times it works nicely because of the people, and some other times it's also a bit more tricky. So I don't know, always the human factor is there still in the yeah. Yeah.
Got it. No problem. Okay, I don't see further questions. I think we're also close to the time.
Top of the hour, yeah.
Yeah, so I guess with that, we can probably close the webinar. But yeah, we have recorded the session, and in case somebody who missed the session, and they can take a look at this interesting presentation. I think the discussion afterwards is also very interesting to me. So with that, thank you very much, Costas, for a great
Thank you.
presentation, sharing your experience with us. Yeah, and thank you everyone for joining today.
Thank you for organizing, Duncan. If you have anyone, you can reach out by email and we can have some follow up. Thanks a lot. Have a great weekend.
Thank you. Have a great
Bye.
weekend. Bye.